When DNA replicates, the enzyme helicase separates the DNA strands. Single-stranded DNA-binding proteins then stabilize the template DNA, keeping it in an extended single-stranded state so it's copied by DNA polymerase. One new DNA strand is called the leading strand, which forms from its 5' to its 3' end by the action of DNA polymerase III. A sliding clamp forms a ring around the DNA and maintains the association of the DNA and the polymerase, allowing the uninterrupted synthesis of many thousands of nucleotides of DNA. The machinery of the replication fork includes helicase and a primase enzyme. Primase creates short RNA segments called primers on one of the template DNA strands. The RNA primers allow DNA polymerase to initiate DNA synthesis along the strand, forming short segments called Okazaki fragments that collectively form the lagging strand. A structure called the clamp-loading protein associates with a DNA polymerase molecule on both of the parental strands of DNA. The clamp-loading protein is also bound to helicase. These associations made by the clamp-loading protein coordinate the simultaneous synthesis of both DNA strands at the fork. Note that the two strands of a double helix run in opposite directions. They are anti-parallel. While the lead strand can be continuously synthesized in the direction of the replication fork movement, the lagging strand template must be folded so that its polymerase can create DNA in the 5' to 3' direction and still move in the same direction as the overall movement of the fork. Using the 3' end of a primer, DNA polymerase III lays down new DNA in the 5' to 3' direction until it reaches the previously synthesized Okazaki fragment. The DNA polymerase then dissociates from the DNA but remains attached to the clamp-loading protein. The clamp-loading protein adds a new clamp and places the DNA polymerase at the next primer. The process repeats. DNA polymerase I replaces the RNA primers with DNA. Finally, DNA ligase links the Okazaki fragments.